I leave the floor to Céline Benini of the Association of Manufacturers and Formulators of Enzyme Products. Céline has a PhD in bioengineering, and she's working at this, let's say, association after her experience also in other private companies. She's now senior association manager in this association. Please, Céline, the floor is yours. And this presentation is about enzymes, a turnkey solution contributing to a better life in a more sustainable society. Please, yes, Céline.
Thank you very much, Davide. So good morning, everybody. So I would like to first introduce a bit our association. So AMFEP was created in 1977, and we currently have 29 members. So as said in our acronym, our members are the manufacturers of enzymes, but also the formulators, meaning those buying the enzyme, making a formulation and putting it on the market. So we have 29 members, which are only companies. And among those members, we have a lot of SMEs, and particularly 10 of them are a very small company with a EU enzyme turnover below 10 million euro. And this is compared to the 2 billion euro production value, which represent the EU enzyme market. Our members represent around 90% of the EU enzyme market and 80% of the international market. So this is actually the skeleton of my presentation today, and those are the key words I would like you to really keep in mind from now on when you think about enzyme. And so the aim here is now that I'm going to explain and introduce each of those keywords. So let's start with protein and catalysts. So enzymes are proteins, which are the primary constituents of all living organism. So enzymes are catalysts, which means that they make specific biochemical reaction happen faster than they would have otherwise. And without enzyme, those reactions would simply not occur or would run too slowly to sustain life. Also, enzymes are classified on the basis of the reaction they catalyze. So it's quite easy. I give you some example, like lipase catalyze the hydrolysis of lipids, protease catalyze the hydrolysis of protein, and it's in general like that you can categorize enzymes. Another important point about enzyme is that they are effective in small amounts, and they operate within a narrow set of conditions such as temperature and pH, hence the little picture about the fact that they can be easily denatured. So another point about enzyme, which is important, is that they are of natural origin. So they come from nature. And so enzyme can be manufactured by extraction from plants and animal sources, or by fermentation of microbial sources. So for microbial sources, you can have fungus like Aspergillus niger. You can have bacteria like Bacillus subtilis and yeast, so Saccharomyces cerevisiae. But you can also have other sources like animal sources, calf, pig pancreas, lamb, beef liver, tongue, and also from plant sources like, as you see there, papaya, pineapple, and barley. So fermentation. So as Jaime explained in his presentation, the majority of the enzymes you will find on the market are from microbial sources. So that's why I am focusing on fermentation. And this is let's say standard process that on the way to obtain enzyme, knowing that, of course, depending on the company, you have slight difference, but let's say this is the standard. So the starting point is the strain. the production strain that each company develop and they maintain in a strain library. So this is really intellectual property, and it's very confidential material that they keep. So all this fermentation work. So there is a previously laboratory-grown inoculum is aseptically added in the first vessel, which is the seed fermentation vessel. And the aim here is to increase the amount of microorganism concentration. Then the total volume is transferred in the main fermentation tank here, which include sterile fermentation broth. So in the fermentation media, you will find, of course, the raw materials such as carbohydrate, proteins, vitamins, and salt. And the aim here is that the microorganism eat this nutrient during the fermentation, and they will produce the enzymes. So you have also to know that the fermentation parameters are really well controlled and managed throughout the fermentation. So whenever the microorganism needs something more, like any nutrient they want more, it's possible to adjust during the fermentation. So after the main fermentation, we have the recovery and formulation. So enzymes are recovered from the fermentation media by engineer operation, which are widely used in enzyme production. So you have centrifugation and multiple type of filtration. And here again, the aim is to separate the enzyme from the production organism and any remaining insignificant residues from fermentation. So then the filtered concentrate is then finished with formulation ingredients, which are acceptable for intended use in order to stabilize and to standardize enzyme. So also interesting to note that yeast and bacterial fermentation can take around 48 to 96 hours, while fungal fermentation can take more than 300 hours. So I should have actually started with this one because this is really the starting point of obtaining enzyme. But so as I said, the production strain is something very important. It starts with that, and so there are several ways for companies to develop those strain. So first, it can be isolated from nature with no improvement, or you can have a classical strain improvement technique like mutagenesis or recombinant DNA technology with homologous strain. So the added genes are of the same microorganism, or heterologous strain with a transfer of gene of interest from selected organism to a suitable host production organism. What is also important to understand regarding the recombinant DNA technology is that the production of enzyme fall in the scope of the content use, meaning that there is no deliberate release of GM into the environment. So, well, now that I explained everything about the production, I think it's time to focus on why enzymes are used. And actually, enzymes are used in a very large variety of industrial applications, such as food, beverages, animal nutrition, textile, and detergent production. So briefly on the technical enzymes, so those used for non-food and non-feed, they can be used in detergent because they break down soil and stain. They can be used in textile for removing impurities and bleaching and decoloring textiles, and also in paper, for example, to remove ink. But of course, here we are in an EFSA event, so better to focus on food and feed. So in feed, they also increase the nutritional availability, nutrient availability for cattle. And I'm sure you have a better knowledge of why enzymes are used in food production. So enzymes are processing aids, meaning they are used during the process. They ease the process, and sometimes they simply make it happen. So they are used in baking, brewing, starch processing, and cheese making. Let's say that those are the main processes. And in many processes, they have an essential use, considering their lack of alternative for the manufacture of certain goods, and also the broad range of sustainability benefit that they can bring. So here, I just wanted to show you briefly the share of enzyme use. So this is a global figure, but yeah, it's more or less similar at EU level. Mainly enzymes are used to produce food, beverages, and also cleaning agents. So this is really the main applications. So the use of enzyme is actually very linked to what they can bring in terms of solution for sustainability and the Green Deal challenges. So enzymes are made from a renewable substance, and they are also biodegradable. They can allow to spare the use of chemical. So example, they can replace surfactants in detergent, allowing reduced temperature in washing machine, and of course less energy use and less release of chemical in the environment. So in detergent, they are particularly useful. They can also optimize food system, like in baking, they can bring better freshness to the product, decrease bread waste. And in brewing and juices, for example, they increase the yield of the manufacturing process. Enzyme can also contribute to obtain healthier product. I'm thinking about asparaginase, for example, that they contribute to the reduction of formation of acrylamide during the food process. So this is clearly lead to a healthier product, and they can also contribute to diverse the food option for notably special diet like lactose-free products, but also low carbs beers. So what is good with enzymes too is that they have an excellent safety profile, and this can be confirmed by the not so many negative or inconclusive opinion that we can find for the moment on enzyme. And this will a bit overlap with Jaime presentation, but yeah, he was more detailed than I will be. So these are the 5 elements that must be considered when conducting a safety evaluation of microbial-derived enzymes, and they are all obviously interlinked. So first, enzymes, they are intrinsically safe. They can be categorized as practically non-toxic, and this is based on over 40 years of testing, use on the market, and in-depth knowledge of their properties. So regarding strain, as I said, it's the starting point, and most and the safety of the production strain remains the primary consideration in evaluating enzyme safety. So, well, the point is starting with a safe host strain and using safe and accepted modification techniques as discussed previously and also in Jaime's presentation. So all genetic modifications are expected to be well characterized and specific. The incorporated DNA should not encode, of course, and express any harmful toxic substances. And then the sequence of the introduced DNA is determined, and the strain is assessed for genetic stability with regard to the introduced DNA and for the potential transfer of genetic material. Then if we go to the manufacturing process, so enzymes are produced using the current good manufacturing practices. The raw materials and the processing aids used are food grade or are safety assessed. And then, as I also explained, the manufacturing process is really controlled. Fermentation is well controlled. It follows aseptic procedure and a specific protocol to monitor growth of the organism. Then we also have quality check for contamination at critical control points. And the final product meets the FCC and purity recommendation for enzyme used in food. Regarding the safety studies, so let's say we have a typical pack, which consists in a genotox and oral toxicity study, which is the infamous 90-day tox. So this is typical. But there are, of course, read across is authorized, and there can be also tox exemption under certain conditions, like QPS microorganism, but also when tox is already done for another strain, which is the same lineage. So yeah, there is the possibility of some tox exemption in very specific condition. And regarding the exposure, so the exposure is calculated as dosage based on the dosage and consumption. And I don't know if you are aware of it, but currently EFSA is building the food enzyme intake model, so the FAME. Well, the industry, well, us, but mostly the downstream user, we are contributing by providing data to the various calls for data. And yeah, so there are already some available, but yeah, some are still to come. So finally, innovation. So for us, innovation is actually answering our customer and therefore consumer expectation. And there is 3 ways of innovating in our industry. So first on innovation regarding the enzyme production or the enzyme profile or the, let's say, extension of use to other food processes. So for the enzyme production, there are out there a lot of genetic engineering techniques, and there are new are still being developed that the industry can use to produce increasingly efficient product, and also to have an accurate characterization of the strain to put on the market also safer strain. So this is a way to innovate. Another way is to find new enzyme. So we would search an enzyme acting on a certain substrate. Then we would look if it exists in the nature and how to produce it, or we can also upgrade the profile of existing enzyme to make them really fit for purpose. And then the last way is on the food process itself. So we are, of course, discussing this with our customers, so the downstream users. We are looking at what are the challenges and issue of the manufacturing process, and we try to find together ways to improve that. And yeah, this is the main 3 ways for enzyme innovation. Oops, sorry. And finally, because this was the title of the session, so the risk assessment at the crossroad of consumer safety and innovation in food production. So our industry is actually responsible, of course, for the product that we are putting on the market, and we invest a lot in order to show EFSA that we comply with the high level of expectation in terms of demonstrating safety. And what is important for us is that we can continue and secure the possibility to innovate. And well, to do that, we think that we need to maintain the good scientific relationship that we have with EFSA, because when we have as industry the knowledge of the production and the industrial knowledge, let's say, while EFSA has the scientific and safety knowledge. So the 2 need to match and to went together in order to be able to provide the safest product possible. And yeah, another point is also to learn from past experience and from the opinion already published. Yeah, as I said, in order to make sure that the new products on the market are really safe and useful. And I think that was, yes, the end of my presentation. And so thank you very much for this opportunity, and I will be happy to answer any question you may have in the Q&A.